Okie dokie. Oh, awesome. We've also got Tom. Hi, Tom.
Hello.
Hello. All
Hey, Steven.
right, everybody. So Happy New Year, everybody. And we are following on with our session that we had back beginning of December to move forward with muscle and neuron modeling here with the OpenWorm project. Thanks, everybody, for taking the time out. I have pasted an agenda, and the link is around. You should also be able to edit on there. So if there are any mistakes or things, please add them. Also, if there are links, please add them. The first page is the agenda for today, and then below are some points from earlier. So I'm hoping that this is going to continue on as an educational session for folks that are trying to figure out kind of what's going on. But there's also been quite a bit of activity in the last month. A lot of you actually doing a lot of stuff, even if it's just asking good questions or running things. So let's go ahead. I'm going to go share my screen, and let's go and look at what we got. All right, so here we are, second meeting, January 3rd. So last time we did an intro to Hodgkin-Huxley modeling. And the muscle model. We had a look at the Boyle and Cohen paper. We had an intro to the Hodgkin-Huxley Python code. We started to talk about this ion channel spreadsheet, and I kind of painted a bigger picture of kind of what the bigger enterprise of neuron muscle modeling with ion channels looks like, and then we touched on LEMS. So if you are watching this and didn't catch that stuff, just check out the previous hangout where we talked about all that good stuff. Okay, so this time I've been sort of piecing together an agenda from all of the various activities that have been going on, and apologies if I missed your stuff. But first big thing to note for folks that are here is that we're playing around with a new chat room called gitter.im, and we've created one specifically for the muscle model. And it's nicely integrated with GitHub. It stores an archive, and everybody here can join it publicly and review the previous archive. Yep, and the link's in the chat. The link is also in the agenda directly if you want to. If you want to click on there. So I've been marking up all these things here with links. So yeah, and right, here's what it looks like. Apparently one of the co-founders of this is also a fan of the OpenWorm project, I learned, so that's cool. Yeah, so anyway, so you can see that folks have already been joining here, and some folks are jumping on now. So this is just a good place to talk about anything related to the muscle model, and you can integrate with GitHub issues just by typing like pound and the number of the issue. So there's good integration with GitHub Markdown. You can get the Markdown help here to kind of see how to format that stuff. There's an integration with the activity on the repo along the right-hand side. So I've been making some edits to this here in the last few hours. So it's just a nice way to kind of catch up on the latest that's going on. Plus you can put in images like Wilson's done here. So you can do @mentions on people's GitHub's IDs, and then also roll over and get people's information. So it's quite nice. This has been a long time challenge for us. We wanted to use IRC but didn't have an archive, and so far this seems to be running well. We'll see if we keep using it, but check it out, have a read, see what you think. Okay, so hopefully we can take this meeting after we're done here and move it on to there and to work out some of the finer points and keep discussion going in the interim. Okay, so we had said for this time that we were going to talk about the muscle model repo itself, and some of you are already working on it, have been working on it, but we didn't, I think, formally introduce it very clearly in the last session. So to go from that sort of abstraction of mathematics and biophysics to concrete code, we have this repository, and I've been working on the README in the last few hours and trying to improve it. And I kind of had to start with like the overview, big picture of what we're doing. So just to refresh everybody's memory, I made this diagram here. So the basic thing. So what are we trying to do here? So we're trying to reproduce dynamics of the electrical aspect of muscle cells of the C. elegans. So here's your super simplistic muscle cell model. It's just an oval. And it's got a bunch of ion channels in here, not to scale, but they're little portholes that allow charged ions to go in and out. Obviously, they actually cover the entire surface, symmetrically, and they're much smaller. And there's a lot more of them, but that's the basic idea. And then by sticking an electrode in that muscle cell, you can make an electrical connection so that your electrode has the same voltage across it that the cell does. And you plug that into an amplifier with a stimulator, and then you can do a couple things. You can, in two different modes, you can either do something that's called current clamp, or you can do something called voltage clamp. Further down in the README here, I've got links out to better definitions on Wikipedia of what those are. But basically, Figure 2, A and B of the Boyle and Cohen paper that we looked at last time are showing you the results of what you actually read out of the amplifier when you're actually plugged into this muscle cell. So you've got a screen, or more modernly, we plug that into your computer, and you get these readings out of either voltage over time or current over time. And in this case, we're stimulating either with current to make voltage or voltage to make current. And these shows you sort of two pictures of the electrical properties of the cell. So this much here is actually not any modeling. This is just the data. This is the experiment that gets done. This is the data that comes out. And this is what we're trying to reproduce with the math that's in this repo. Okay? So hopefully that much should have been clear in the last time, but this is, I think, a nicer summary that's just kind of all together. And the fact that these are all overlaid, you'll remember, is because we do a bunch of repeated trials. They're all overlaid on top of each other, so it's just sort of showing you stimulations at different levels and what comes out in the current clamp mode. Same thing here with voltage clamp. It's sort of setting the voltage at a level for a period of time and then turning it off, and they're all overlaid on top of each other. So in this repo, then, this is where we have different versions of the mathematical model that tries to reproduce these dynamics. Okay, that's the whole point of what we're trying to do. And so what we've got in this repo are approximately 4 different things, 4 or 5 different things depending on how you count. We've got a simulation, the original simulation from the Boyle and Cohen paper that we were looking at. So we actually got Netta Cohen from Leeds to donate their code to the OpenWorm project, and that's what's under the Boyle and Cohen 2008 directory. And then in addition to their original code, we've been adding a MATLAB version of some of that into this repo. So the original version is in C++. You can see all the C++ that's here. And then there's also a MATLAB version that shows a lot of the components, so folks can run in MATLAB. But in the OpenWorm project, we prefer Python because everybody can get a free copy of Python, very easily to run it. And so graciously, Rainer Lucas and Vahid have been contributing to porting pieces of this over into Python, and that's what's available under here. And in the README recently, I've just added, this is a screenshot of running vclamp.py. So everybody here should be able to run vclamp.py as a minimum sort of level of kind of understanding what's going on. And hopefully you can see, more or less, that vclamp.py sort of looks like figure 2b from the original data. Now, it's missing this original spike, and I think Miguel asked about this earlier. That is the way that the model kind of ends up. It doesn't like have that big obvious spike at the beginning, and we can talk about why that is. But essentially, it's what the authors of the paper kind of didn't have it in there originally. So we're supposed to at least be happy that it captures the dynamics that the slower dynamics that happen after here. But it does seem to sort of miss that original spike. But okay. So that's the first thing that's under here is the original, and the purpose of having the original is it's good because then this is the the ground truth, or the basic standard of what you're trying to reproduce. So now we're kind of, so there's the original data, which is here, and then there's the Boyle and Cohen paper version of that, and then there's the Python port of that, right? So we're 2 versions away, but at least the port is a pretty faithful port as we're seeing it. And we probably also want to, at some point, get the iClamp version, which is going to reproduce Figure 2a, ported over. That is available on the MATLAB side. Let's see, we're getting some questions here. Okay, Joe asks, is there a way to run the MATLAB on Linux without owning MATLAB? Right, good. Abdullah, yes. And Travis, yes. So Octave does work. Rainer, you were running the MATLAB in Octave. Do you want to say a little bit about that?
Yes, yeah. When I was porting the vclamp script, yeah, I was testing it all out in MATLAB to in Octave to find out exactly what the MATLAB scripts were doing, and yeah, for the most part, it just worked. So
yeah, exactly. Yeah. So we're gonna, we try to avoid heavy, use of MATLAB because there will be places where Octave will not do all the fancy things that MATLAB can do. But it does look like for these simple scripts are sort of self-contained, so you can go back and forth and look at, vclamp.m versus vclamp.py. I think they don't use super fancy MATLAB features. So yeah, you can give that a shot. It'd be pretty interesting probably for folks. Right. So anybody want to say anything else on that? Okay. All right, cool. So so this was the basic idea of to get the running code, which is sometimes challenging, by the way. So it's very gracious of Netta Cohen to have given us the original code, because all too often all you're going to find is just the graphs and the equations and the parameters in the paper. But actually having the code itself is usually pretty critical for that. Okay, so that's the first thing that's in here, is that stuff. Then there's also been a conversion into Neuron, which I haven't written as much about in the README, and I'm also not so sure that the Neuron implementation README, yeah, so this is still in need of some improvement, quite a bit of improvement. But basically, this very simple thing was added, I believe, by Mike Vella originally, and although it looks like it's just a Python script, it does use something called Neuron. If you're not familiar with Neuron, let me just really quick introduce it. So Neuron is a general purpose simulator. It is a classic simulator. It has been used in hundreds upon hundreds of papers in the field of computational neuroscience. Like, the bibliography just goes on and on and on and on. So you can check it out over here. It is a workhorse of running simulations in computational neuroscience. We have a complex relationship with Neuron in the OpenWorm project. It is this workhorse, and it does have a lot of papers published on it, and so there's a lot of credibility and a lot of code that's been written in Neuron. However, it has some challenges for sharing in open source. It uses very specific languages that you kind of have to learn very specifically. As you can see from our repo, if we go back, basically the way that you make a model in this is you use these .mod files. These .mod files are kind of its own language that was only ever used for Neuron itself. It's a little obscure. It doesn't have a lot of type checking and these kinds of things. So while Neuron is useful, it's also complicated to use. The GUI is complicated to use. There's a big learning curve in getting to use it. But some of us in the project have learned how to use it and gone through tutorials, and there is a book that you can read to learn a lot about it. And it is instructive if you're curious about it. But for the moment, we're trying to kind of move past Neuron and use some of the newer technologies, because They better enable sharing. I think they better enable open source work, and they enable a lot of checking and testing. Basically, another challenge I should mention is that, right, so Neuron kind of operates as its own application. It's not a library. So even though this Python file here is using Neuron, sort of importing Neuron, this is a sort of a long time in coming of a kind of a Python wrapper around Neuron, which is pretty challenging to build. You can build it, but you know, on different systems it builds, you know, more or less easily. It's probably easiest to build on Linux. It's pretty hard to build on Windows. On Mac, it's like okay to build. But basically, yeah, so running this script kind of has a prerequisite of having installed Neuron just right. It doesn't install automatically. You generally do have to build it from source. But that is in there, and like it says, yeah, this is sort of not under development as of, you know, a couple of years ago. Development focus has shifted to kind of some other stuff. So, but Mike did have this in there, and you know, it was a nice start, and so it's kind of in there for archival purposes, and you feel free to play with it. Okay, the second thing that's in here is, I think, the thing that we're really more focused on these days, which is NeurML2 conversion. Let me see what's going on here. I tried installing Neuron, but gave up after compiler errors and every prerequisite. Yeah, right. So like Wilson said, yeah. In the early parts of the project, like, you know, we recommended folks to try and install Neuron, and we just found, like, it's just really challenging. So yeah, that's the issue at the moment. Okay, so NeurML2 LEMS conversion of the muscle cell model. So the reason we like NeurML2 built on top of LEMS, I started to talk about before, is it has this nice property of you know, describing components of the model in a declarative form of XML. And so the second thing that's in here is that we're sort of moving this into what's under NeurML2, and there should be a direct link to that in that part of the README, but there isn't. But anyway, that is under here. So this NeurML2 part. So this is the second bit. C. elegans muscle model cell in NeurML2. So there's instructions on how to get it, instructions on how to go to it, and then you should be able to run these two things, although I haven't done it recently. But you should be able to run these two things, and these are going to, again, reproduce figure 2a and figure 2b. But you do have to install this JNML package, which sometimes is a little bit of trouble. It is a Java thing, so you're going to have your Java install in your system working correctly and pointed and all that good stuff. But so again, figure 2a and figure 2b are these two figures. So we have this next representation that should be showing that stuff. But the thing is about this, you know, .xml file is that it's this declarative form. So when you separate the implementation from, you know, some piece of code, now you have this key property of composability, which we're really trying to get to, because it's not enough for us just to reproduce this muscle cell once. We need to clone this muscle cell 95 times for all the 95 body wall muscle cells, and they all need to kind of be able to work independently, and we don't want to be like copying and pasting code. But with this nice NeurML2 language, we can instead, you know, indirectly reference our muscle model 95 times and be able to, you know, modify it independently and test it. And so there's a lot of good properties that come along with this. So that's the second thing that's in there. And this is still in progress, but has been making, you know, additional progress recently, and has been the main sort of focus of work that, you know, we started, that Rainer and I have been working on with Porig. And so I think a second thing that you should be able to do is to be able to run this as a minimum prerequisite on your own. Now, in the midst of talking about this NeurML2 thing last time, we got into, well, what is really going on behind the scenes? And so, okay, yeah, so run vclamp.py. We looked at vclamp.py, so that's one thing to do. Yeah, running the NeurML2 examples should be another thing that folks should try to do. And then, okay, so what's really going on? So Joe Bowen has done, oh yeah, so this is just here, by the way, saying I'm continuing to update the README to add more instructions on how to install all that stuff. But so Joe, you've done a really nice job with this Hodgkin-Huxley NeurML LEMS tutorial, and I had my own thoughts about how to walk through it, but I thought I'd give you a chance now to just kind of talk about it and your experience of how to do it and what you did and what motivated you and how you feel it is going at the moment.
Sure. I think part of the trickiest part about it was really getting, almost getting JNML working.
Okay.
And eventually I got it working. There was a .travis.yml file. It's not really an install file, but if you read the actual steps in the file and run those commands, it seems to install everything properly. So that I think I'm talking about the same thing.
What, JNML, right?
Yeah, I think that's the issue I was having. I think. So once I got that installed, it was really just finding there was an existing Hodgkin-Huxley model that was in that Neuron ML format, and I could kind of piece together between the Python file and that file to kind of get the correct settings, I suppose. I'm still missing 3 initial values and kind of how to set those. I think we talked about it, but I think I lost the email.
Yeah. Okay, so hang on. So let's just start big picture, right, of kind of like what you did. You want to just tell folks, like, at a high level, like, what the idea is here? Maybe just starting with what this Hodgkin-Huxley.py was?
Sure. That's the Hodgkin-Huxley .py. It forms a few couple example graphs. The pictures over in the getter.im of what those graphs show. But then, yes, right there. And then basically the idea is to output the same thing in a in the new neural ML file format, or model format.
Right. Exactly.
So, yeah.
So last time we talked about this Python file. Basically, what this guy does is it forms the nonlinear differential equations that compose the Hodgkin-Huxley model. It sets a bunch of the parameters for those equations, and then it uses matplotlib to plot out those dynamics over time. And this is kind of the first intro into going from the math that I talked about before to some code that actually works. And then, and so let's talk a little bit about these 2 files then. So these 2 XML files, like Joe said, are examples that have been produced by the NeurML2 folks, and they also have simple Hodgkin-Huxley cell dynamics. But here it's done all in XML.
Sorry.
Yeah, go ahead.
So now the dot, which file are you on right now? The
I'm on
the .xml file actually describes the type. Let's see here. It describes the actual
simulation
output simulation, right. And the other one, the .nml file, kind of describes the cell itself.
The cell itself. That's right. That's right. And so that's already showing a little bit of the decoupling that's nice about the system, which is that here this is just a cell independent of any instructions of what input to put into it or what to plot out or any of that. Okay, so this just has the rates, what ion channels should be in there, the size and shape of the cell, properties of the membrane, etc. This file over here, this XML file, talks about how long to run the simulation for, what step size to use, what graphs come out. Whereas in the Hodgkin-Huxley.py, all this stuff is all kind of in the same script file, right? What you plot out and that kind of stuff.
Right. If I want to combine multiple cells, I would just call the cell or the .nml file several times and map out the the interconnections. So
Exactly.
it makes it pretty easy to go from one cell to a network.
Exactly. Exactly. So so essentially, so now turning our attention real quick to let's see what to do next actually. Okay, well, so the other thing to note is that yeah, so what Joe has done is he's gone line by line basically comparing those files together to show you like this thing in Python is this thing in NeuromL2. You know, these parameters in Python are kind of over here on this line of the NeuromL2 file, and kind of going back and forth. Yeah, I think he's done a nice job of showing you like where those pieces are. Like this thing from the Python goes in the XML file defining the simulation, and it kind of goes all the way through that stuff. So it's very nice. Yeah, showing you how the plots work and that kind of thing. What I was suggesting in the Gitter chat room was that actually that part that you said that was hard, which is the installing of JNeuromL, would actually be nice for us to kind of put in here somewhere.
I think we can pretty easily do that.
Yeah, cool. And since Wilson produced this graph, I was thinking he could do it as well. But either of you, if you get a chance, I think it would be excellent for folks to kind of be able to get in there.
Actually, what I'd like is I could write up what I think is it, but I'd also like someone who hasn't actually installed it yet on their machine to kind of run through my directions and make sure they're actually right.
Good. Yeah, I think we can find somebody in the room here today who can serve that role. So awesome. Okay, so then, so really great contribution, by the way, Joe. So this is a great initiative. So really glad about this. And whenever if you ever want to put it under the OpenWorm organization or whatever to kind of give it more prominence, just let me know. That would be awesome as well. But that's up to you.
I'm all for it. We can talk about it after this conversation.
Cool. Awesome.
Yeah.
Okay. Also, I guess parenthetically, one thing I should say about the form of this that you picked up on is you kind of got your first intro to read the docs, right?
Yes.
And formatting, how that worked. So this is so just for folks on in general, this is backed by a GitHub repo. That is nicely editable here, and
it's beautiful.
Yeah, and
it's so beautiful. I've actually integrated it with about 3 or 4 other projects right now, so I really like it.
Yeah. Okay. Good. So yeah, and actually, did you want to comment on like RST versus Markdown, because that sometimes is a bit of a sticking point for folks. Or initially, I know you were asking about that. How did you find it's the restructured text Markdown versus the just like GitHub MD Markdown?
I think I just picked one.
Okay.
Maybe might have been the tutorial I followed to figure this stuff out, but I really just kind of picked it.
Okay.
There wasn't. I liked it. I mean, it makes citing things very nicely. You can cite things by line number. In Python, you can actually even cite things. You notice I'm citing the languages and the line numbers, and you can also even cite things like functions, at least with Python.
Yeah.
Makes
awesome.
It very nice.
Yeah, so this is what it looks like behind the scenes, by the way. This is like the raw source of the tutorial, and yeah.
Now, one thing I'd like help with is kind of sprucing up some of this language. I'm not a very good wordsmith.
Yep. I can also help with that.
Probably kind of describing what these things actually are in terms of the Hodgkin-Huxley model itself, versus just like plug this into here and plug that into here.
Yep.
I'm thinking.
So, yeah, no, it sounds good. I think you should, like, all those ideas you should jot down in issues, actually.
Okay.
And we can kind of note all those, because yeah, each one of those things is good. Probably also the one that I was thinking about with adding the JNML stuff. So anyway, yeah, one of the important things, in the project in general, and that Joe's done here, is that we always have to be thinking about how somebody else is going to see this stuff when they come to it. It's one thing to have the models up there, but it's another thing to know that people are going to be able to like pick it up and use it. And so that's why I just kind of wanted to make this aside real quick here on like the methods of writing good documentation. basically any repo that uses RST, you can just point to read the docs, and this came up, I think, probably in like 5-10 minutes of just like setting it up, right? It's pretty straightforward to use once you do a little bit of the investigation on it. So the more that we do this, the better. OpenWorm itself has its docs using the same setup. And in fact, what I'll probably do is find a way to link directly to this Hodgkin-Huxley tutorial from here, or even like integrate it under the subdomain. I don't know. There's a few different ways to do this across repos, but so anyway, yeah, this is quite nice. Okay, unless there's any questions about any of that. Okay, so let me keep going. So I think the main thing to understand about the muscle model is that it's essentially a modification of this template of a Hodgkin-Huxley type model. Okay, so like this single comp HH cell.nml, you're going to find a lot that's the same when you look into the NeurML muscle model. It's written in the same language. It's going to use a lot of the same terms. What's going to be different is that there's different ion channels. So instead of an NaChan, which is a sodium channel, there's going to be a calcium channel. There is going to be a potassium channel, but there's actually going to be two different potassium channels, one that's slow and one that's fast. And what's different about them as well is some of the parameters that are in there. But fundamentally, it's using the same system of equations under the hood. So you're just kind of plugging and playing, these different ion channels, maybe some of the, of the passive properties of the membrane, these kinds of things. But essentially the form is very similar. And I haven't actually looked at it in a little while myself, but let's just see if we can actually just kind of line these things up in here. So, so we said, running figure 2a and 2b. So we're going to expect that the .xml files are actually not going to have the model in it, right? Because as we learned, this is just giving us the plotting, telling us what we're going to display. But what we're sure to find when we look in there is that there's an include for some of the stuff that makes it up. And in fact, it's referencing all the different components. So with the .nml extension, you're seeing what the components of what the plot that comes out here are actually made up of. So what we can see is that single comp muscle.cell.nml is one of the things that's getting referenced and looks pretty likely to be the muscle cell. So now if we look in here, we can start to see some pieces like biophysical properties. And let's see, is this? Yeah, this is NeurML too. Channel densities, intracellular properties, and I guess, let's see, okay, so I guess the ion channels themselves are separately referenced, so K-slow, K-fast. So in the Hodgkin-Huxley example, they're all put together in the same file, which is very nice. Here, we're going to have the ion channels as separate XML files, but you can think of these things just being copied and pasted together. So here's the ion channel for K-fast, whereas over here, it was, this is the ion channel HH K-chan. You can see one of the differences too is that there's these pre-made Hodgkin-Huxley ion channels that are pretty standard under the tag ion channel HH, whereas over here, it's just ion channel. And so it gives you some more flexibility in specifying channels that don't follow the exact HH pattern. So there is documentation for the difference of those things under NeurML 2. I don't have a quick pointer to it right now, but I'm sure if you're curious about it, I can point you to it. Basically, this is all under GitHub repos. I just don't have the link immediately, but each one of these tags are essentially described and defined in the NeurML 2 schema. XML2 schema being a key technology that's being used here. So all of these tags have some definition that's also described in XML that you can go back and see every single attribute here, like what is this conductance, what is this species? Well, it's all described in the docs for the NeurML2 schema. Let's just look real quick, actually. NeurML2 schema. Yeah, so it should be under here, and there's probably some fancy place where it's all rendered that I don't know. Anyway, you've seen the paper, a lot of those block diagrams describing kind of what you can describe in LEMS. which is the foundation of NeurML2, is showing you some of that stuff. And let's see, NeurML2 schema. Yeah. So somewhere this is all sort of self-described, but the source of it is under here, where essentially every type is defined. So like ion channel. Oh no, let's see, ion channel. So yeah, ion channel. Oh, so it turns out ion channel and ion channel HH are currently functionally identical. This is needed since many existing samples use ion channel, some use ion channel HH, and in the next version they're planning to remove some of these. So you can get these kinds of definitions under here. And but anyway, hopefully you don't have to read XML schema too much to do this. There's also some more human-readable ways to look at NeurML2 schema. Okay, so the muscle model is a specialization of what you see here. So if you understand the Hodgkin-Huxley LEMS files, you're really close to being understand the LEMS files for the muscle model and being able to run them. Okay, line by line. Next bit, running both. Yeah, so I sort of maybe flag Wilson or Joe for the next version of being able to like have instructions for folks to actually run them on their own system. Okay. Generating IV plots. Okay, so we've had some progress on this as well. So Vahid has opened this issue and helped us work on this. So there's these two different versions now of generating IV plots for ion channels. One version is under the Boyle and Cohen paper directory that we talked about, and so Vahid has put this together in the last 10 days. A really awesome contribution as well, where he's basically taken the Python Hodgkin-Huxley.py and modified it and plugged in essentially this this muscle model code and tried to generate the IV plot itself. So, and the other version is coming from work that Rainer's been doing, which is not just ivanalyze.py, but is actually a couple things. So let me explain first what an IV plot is. You should remember we talked a little bit about it last time. It is in the Boyle and Cohen paper. Let me just reshare that bit. But basically, I stands for current and V stands for voltage. So it's a plot looking at the electrical properties of a given channel. You can do an IV plot for a whole cell, or you can do it for an individual channel. Since the individual channels are what make up the dynamics of the whole cell, it's important to know that your IV plots are going to match what the original data is. So if we go back to the paper, so figure 3 of the Boyle and Cohen paper were these IV plots, otherwise known as current voltage relationships. I for current, V for voltage. So current voltage relationships or current voltage plots. And this follows the same pattern as figure 2, where there's real data for A and B and simulated data for C and D. And so A goes with C and B goes with D. The A current voltage relationship is for the calcium channel that is within the muscle model, and the one in B is for, I guess, all potassium, which merges, I think, the K fast and the K slow into a single graph. The way you generate this experimentally is that you will do you will do a current clamp or a voltage clamp. Actually, I don't remember exactly which one you do for this. And you'll do one of them, and then you'll set current at 1, and you'll see what the voltage is, and then you'll set current to another value, and you'll measure what the voltage is, and you'll set current to a third value, and you'll set what the voltage is. You'll measure what the voltage is. So whereas in this picture we're actually seeing the whole trace, in a current voltage relationship plot, you're going to either pick the peak, as it does in one case, you're going to pick the steady state, as it does in another case. You're going to measure that value, and you're going to generate a plot like this. Current voltage relationship plots are something originally from electrical engineering to define like a device. So you can generate a current voltage relationship for a resistor or a capacitor, and it's kind of like a fundamental electrical test to see what something does. And for ion channel electrophysiology. It's been imported as a way to kind of get a unique fingerprint for the dynamics of an ion channel. And one of the key things is that there's no time in here, right? So for figure 2, we're looking at how things are changing over time, and it's very sort of sensitive to differences in time. But a current voltage relationship doesn't have any time in it at all. You take the time component out. And so it's this like snapshot kind of the dynamics of the ion channel. So the idea is if you get these right, then you're going to get this right. And mathematically, the dynamics of the ion channels are kind of added up together into the dynamics of the whole cell. So questions on that? Kind of touched that before. Okay, so what we've been doing is we want to do the same thing with generating the dynamic plots that we've seen in Python by itself. It's what the Neuron code generates, and it's also what these figure 2a and figure 2b plots do. We want to do that for current voltage relationships. And so these two different implementations, one of which is very easy to run now, that Vahid has created IV all, and the one that Rainer's created, which is we're still, I guess, we're still working on how to get it easy to run, are the two main things. And you can understand why, as we're moving to the NeuromL2 implementation, what we'd like probably most is to be able to run current voltage relationships using the NeuromL2 LEMS implementation directly, because that gives us the maximum reusability and shareability. But at the same time, having the pure Python example also shows us the ground truth based on the implementation that was closest to the original model. So these are still in progress, but are getting better and better. Now there were some additional, I think, topics on this to kind of discuss. So I don't know, Vahid, can we hear you? Do you want to say something about your IV all implementation?
Yes. Hello, everyone.
Hello.
What I was doing was based on the ion channel spreadsheet we were working on, and if I can share my screen.
Yep.
I was working on something like this to finally generate some ion channels that can be generated from something like these literatures, and we can complete that spreadsheet from these literatures. The code I was working on was something that you explained, and I finally could generate 4 curves. That my last script was generating these 4 ion channel curves that are calcium, potassium, the 2 kinds of potassium, slow potassium and fast potassium, and finally the integration of these that is the final action potentials that are generating for the muscle cell. I was working on that, and mainly on the work that in project has been done. We have
Oh, still there?
we have this finally extract our models, something like this, that can be used in our model. I'm working on this one, and also something that I was working on was some tools that we can use to digitalize the last parameters that we want from literatures that's something like this tool I found very useful, that you can capture the temperatures, and just by matching the points we can finally extract the data we need from the temperatures. If we don't have the data from many literatures that we have just some figures, we can extract the data finally, and something like this we can have these to to put them together and to have the last equations for our models in Hodgkin-Huxley model. These were the works I was working on, and I hope I can finalize them in near future.
That's awesome. Can you share a link to that digitizer as well somewhere? Either is that like free software somewhere, or yeah? So the basic idea, guys, right? Yeah, that's really cool. Yeah. So the basic idea, right, is that so this spreadsheet which we talked about before, there are these families of different ion channels, and there's papers like the one that he just showed the picture from, where they've characterized the IV plot of that given ion channel. So then it's up to us to be able to have code that can generate an IV plot from a model that we've described ourselves, and make the picture that we generate and the picture that's in the paper match. So what's cool there is that he's got a digitizer where he's taking the picture and he's actually finding a way to digitize what the curve is, so that we know what to match to. And then the next piece that's challenging, which I think there was some discussion about how to do it, is, okay, well, now how do you pick parameters for your ion channel model that are going to match that? And that gets into the topic of optimization. And I think I have other things that I kind of wanted to talk a little bit about before we get there. Oh, we're making actually pretty good time. I do want to talk about C302. I don't want to go too much into optimization right now, but if we have more time later, we can talk about that. But basically, yeah, the trick, so you can use fancy AI techniques, essentially, to take your parameters for the model and the target and match them together. And there's been work by Mike Vella, who was previous contributor to the muscle model, in a few different packages to do this, and some folks here have been pulling that code out and trying to get to work again, and have had some various successes. But the basic enterprise is to have that, and also have it integrated with the LEMS and the NeuroML thing. So this is the last bit, right? Which is to say that, like I pointed to before, those ion channel blocks of XML is kind of the gold standard, is what we want to have for each of those ion channels. So we basically want to build up a library of .nml files that have ion channels in them that we know are matching papers. And even better, those ion channels are going to reference the paper that it comes from. It's going to say this is taken from this figure in this paper, right? So that anybody who comes along can say, well, why is that ion channel? Like, why do you think that's the right model? And you can say, it's because it comes from that paper. And this is the process that was used to fit the parameters to it. And you could even have a unit test that reruns the optimization process so that everybody can see, and it's very transparent, like where all those parameters came from. This is kind of the enterprise of what we're doing here, and so Vahid has taken that step. So I think the thing is that we want to, so it's good to have the IV analyze where we just plug the parameters into Python code, but even better is to run them under this tool chain of the NeuroML2. And so Rainer, so to you, Rainer is the person who has done that, not the optimization part, but the process of running IV analyze scripts on NeuroML2 files. Are you still there, Rainer? A little frozen. Yes.
Hi. Yeah, still here.
Cool. So I kind of flagged myself as the creator of the steps to do that in the last session. And what I've only had the time to do is to kind of put the top level intro pieces together and kind of clean up that README. But can you just sort of basically explain for folks, maybe just right now, like what are the steps? Like if somebody here wanted to create an IV plot, or like run the IV plot of the potassium channel or the calcium channel, like how do you go do it? I know that Vahid has done it for the potassium channel. I think he ran into a problem with the calcium channel, and I think that had to do with the install being complicated. So do you want to kind of explain that? If you want to take a few minutes to put that together, so that I'm not just putting you totally on the spot, you can. But you're the go-to expert for that. So.
Sure. Okay. First of all, what you'll need is the muscle model repository and the Blue Brain Project Showcase repositories installed from GitHub, together with all their prerequisites. And
so actually, hang on. So the Blue Brain Project Showcase is something we haven't talked about before. So we should probably flag that. Here's sorry,
go on.
No, here's Rainer's version of it. I'm just linking to it, and I'll also share what it looks like here. So you may have heard of the Blue Brain Project. It's the precursor to the Human Brain Project in Europe that has gotten a lot of press, and this is kind of an intersection between the NeuromL2 world and the Blue Brain Project world that is sort of showing how to pull in some examples of individual channels. This same strategy that we're using here for C. elegans is, in fact, the same strategy that they use for getting good descriptions of individual ion channels for cells. Only they're doing it for human cortex with billions of cells with a whole lot of ion channels, a whole lot more ion channels. Actually, couldn't tell you what the number of different types of ion channels that they would need to have. But I can tell you that there will be somewhat less, maybe not exponentially less, maybe just linearly less, but there will be fewer ion channels to figure out for the worm. So that's always one of the exciting things, is that we are kind of piggybacking on the same strategy that other major projects are using, but doing it for an organism where it's much more kind of constrained. So this repo here, which Rainer has cloned, has started to show what's possible using NeuromL technologies and using some of the examples of channels that none of this is worm specific, but some of the channels that they have encoded themselves. It should also be pointed out that there is a precursor to NeuromL2 called ChannelML. ChannelML is a NeuromL1 thing, and it allowed you to describe channels. The NeuromL2 version of channels is just NeuromL2, so it doesn't have a special name. But if you see something that's called ChannelML, that's NeuromL1, and it's a little bit different. There are some converters to go from ChannelML to NeuromL2. But okay, so the idea of this repo was to start to build up these tools to be able to use NeuromL2 and do stuff with channels. So okay, now having said that, back to Ring.
Okay, so yeah, once you've got those, you'll need to get those two repositories installed with their prerequisites, and I believe Joe was talking about this earlier, just how to get these installed, what I've just been doing earlier today was just writing up a list of installation instructions, because I don't think we have one yet. So yeah, I guess sometime later today I can send that. I can send it as a pull request to go into the README for the muscle model repository. That's probably the place it makes most sense to put it.
That'd be great.
Okay.
That works. I actually put an issue in there, so there's an issue for that.
Oh, cool. Yeah. Okay, thanks.
Yep.
Okay, yeah. Okay, so once those are installed, in the muscle model repository, there's a NeuromL2 directory that contains ion channel descriptions and also some little shell scripts that that basically call a channel analysis Python script from the Blue Brain Project Showcase code. And what this does is it takes one of the ion channel descriptions and creates a LEMS simulation.
Okay, so hang on. So I'm going to just kind of walk through this a little bit more step by step, as you're saying. So, okay, so at the highest level, there's this test.sh. And underneath, so what's happening is it's going to run a few things. It's going to run JNML on all these guys, and as well on the figures. Okay, so the first step of validate, this is just a pure test step, right? This just basically says, are those NML files sensible? Do they make some sense, right? That's pretty much your understanding, right?
Yeah.
Okay, yeah. And then these guys, these JNML, are going to generate all right, so these are going to actually execute the simulation under that XML, under these two XML files, and should produce some data files, right?
Yeah. Okay, those are two example simulation files that are supposed to replicate figure 2A and 2B from the Boyle and Cohen paper.
Yep. And Joe asks, yeah, can we turn the Travis into an install script? Yeah, I think we should.
That would be good if we. Yeah, that would be a good thing to do.
That'd be very helpful. I know that sometimes the Python packaging can get tricky, though, from personal experience, so they can be slightly painful. But yes, I think that it would be, if we can do Python, or like pip install on this, that would be brilliant. I think we've tried to do something like that, but I don't think we've gotten very far. Okay, and then there are these other. So then these three scripts you were starting to talk about as well, right?
Yeah.
And so, like, let's look at analyze case flow .sh and what's going on there. Okay, so what these three are doing is that they're going beyond just having run the simulation for these two files, or maybe they're running independently. Let's see. The first thing is that they are running NML2 channel analyze.py, and this is one of the key bits from the Blue Brain Project Showcase. And so I guess this is yet running another simulation because you're putting in a bunch of parameters.
Yeah, I guess creating the simulation file so that we can run it using Neuron.
That's right. So that's the first thing it's doing, is it's creating, and I guess it's going to create this .xml file, even though it's possible that this appears in the repo already, but it will generate this new LEMS file that then, when you execute JNML, will run the full simulation. Yes.
Yes.
Good. And that produces .dat files, right?
That's right. Yes, it'll produce a set of graphs if you run it just straight from the command line, showing things. A number of different variables, but things like current through that ion channel over time.
Can you do that, by the way? Do you want to show that? Is it easy? Do you have it set up, not broken at this moment in time?
Yeah. Just let me see if I can share my screen.
Yeah, because it's a pretty impressive set of graphs. It might actually be worth talking through what they all are, just really quick at a high level.
Okay. Yeah. Sure.
If you haven't seen this, this is good stuff. This is like. I get excited about this stuff. It's kind of the future. This is like digital electrophysiology. So for somebody who studied neuroscience, these would be tests that you would do in a painstaking way with an amplifier and over time, making measurements and this sort of thing, and they all just get generated for you. So it's like everything that you want to know, system identification about an ion channel, just poof, like all run and simulated for you. It's like, so we're kind of plugging you into this like in silico ion channel experimentalist setup. It's like this is just going to look like a bunch of graphs to you, but I think it's like really cool that we can get to that level of precision. This is the thing that for biologists I think is where the rubber really meets the road, because these are literally the same graphs that if you're studying the ion channels you would need to generate. Sometimes you can't even get these graphs because the experiments take time to do. But I really like this stuff, and it's important, and it's important that it's all there because it's the proof that we're in fact reproducing biology and not doing something abstract. Okay, so it looks like if we look over at Rainer's screen.
Okay, yeah. So I'm just running the in that analyze CA Boyle script manually, mostly because I've just got it installed in a different directory, one where you expect it to be. So the first step is just to run this NML2 channel analyze script together with all of the simulation parameters that we want it to have, and pointing it at the channel description file and telling it the name of the ion channel. And what this does is it writes out a LEMS test file called LEMS test and name of the ion channel.xml. And what we can then do is run that simulation using JNeuroML. And if I've got this set up right, this should produce a bunch of graphs now.
Fingers crossed. And by the way, so CA is calcium, and underscore Boyle is that it's the calcium channel from the paper.
Yeah. Okay, so we've got a. Okay. This is probably our most important graph that we want to have a look at. That's the current through that ion channel at various different voltage clamp potentials. So that's current against time. Red line is at a membrane voltage of 75 millivolts. Lightest yellow one is minus 55 millivolts. So, yeah, and I can zoom in on that slightly.
Yeah, and just make a connection there. That's the same range. Let's see, from what, minus 70 to, oh no, minus 55.
Minus 55 to plus 75.
Yeah. So within that range, you get the ion channel here. Hang on, let me just share real quick. So in the paper, right? So this is just to draw the direct connection. So this is supposed to be a model of Figure 3A. Figure 3A's content, except what you're seeing is the graph over time, and here this is the one without time, which is the current voltage relationship by itself. But in order to generate the current voltage relationship, you need to test what current comes out at a given voltage. And so this is a bunch of experiments of holding it at different voltages, seeing what happens to the current, and from that you can then pull out this IV plot. So you have to kind of have it over that range, and that's why we're doing it that way. Yeah.
Okay, and yeah, this also gives us a bunch of other useful graphs. This one's just voltage against time, so it's just showing what all the different clamp voltages are and the times when the clamp is on. Yeah. Okay, channel fractional conductances and activation and inactivation variables. I'm not sure where the oh, right.
So we didn't talk a lot about activation and inactivation variables in the first discussion, but if you got into the weeds of the equation underlying the major the top level equation, which is the capacitance, and it has the big voltage term in it, is the sum of the ion channels, and under the individual ion channels there are these activation and inactivation variables. If you go through the tutorial in the Python code and in the LEMS code, you'll see those gates described with letters like A, B, C, D, or M, N. Usually, classically in the Hodgkin-Huxley, it's M, N, and in the paper, it's things like E and F for the calcium channel and those sorts of letters. So these activation and inactivation, and those graphs there that he's showing are the time course of those things either opening or closing. They're basically from 0 to 1, so they're just literally like how open or closed is the channel, and then what it's showing you is like over time, like how fast is that going? Does it close really fast, or does it close much more slowly? And depending on that dynamic, that you could imagine that that's going to also have a bigger impact on how the whole channel functions and how it relates to voltage and that kind of thing.
Right. Okay, and so what this should also have done is created a bunch of data files.
Right.
So if I just... Okay. So it'll save files with all of the simulation values for the activation and inactivation and time course variables. I slightly modified the template file that it creates so that it also saves a list of currents at each simulation time step for each clamp voltage. So basically, this is data for each of the lines on the current graph that we just saw.
Exactly.
So yeah, well, I'll just look at one of those. But yeah, it's just time in seconds and current. So yeah, I also created a little Python script that's just IV analyze, and what that does is just processes all of these files with the current values in them, looks for the highest current, lowest current, and absolute peak current, and plots each of those as a graph. So I'll just try running that on the files it's just created.
Okay, so one thing here that we've been going back and forth on chat, and just want to see if we can resolve right now. Right. So Vahid has tried to run this and has been able to run the potassium, hasn't been able to run the calcium one that you just showed, and it has something to do with the template that doesn't have the output files or something. Is that something that we can just like have a quick sync on?
Yes. Yeah, those template changes only exist in my fork of the repository at the moment.
This is the thing.
They're not merged with the main Blue Brain project showcase.
Your fork of Blue Brain Showcase, right? So this link that I gave before, that's the Rainer/Blue Brain Project Showcase. Vahid is the one that you want to use. Have you been able to use that one?
Okay.
Well, okay. Maybe chat it or something. But anyway, okay. That might be a key point for everybody here when trying to run this. Again, we're Rainer's working on the instructions to get all this working, but that's a gotcha point that one folks already run into. Okay.
Yeah. So for that, I'm not really sure whether it's worth submitting my modified templates to the Blue Brain Project Showcase maintainers, or whether we should have an OpenWorm fork of that repository that's for the changes we need.
We could do that. We could do that. Yeah.
I mean,
then they're free to pull it into theirs if they like.
Yeah, I mean, would that be a sensible way to do it?
Yeah, I think so. I can make a note to pull in that. I'll fork your version basically into the OpenWorm repo, and then then we'll have the changes because then there they will be, and then we can write the readmes pointing to that instead of to the one under Open Source Brain.
Yes, yeah, that might make sense rather than just having it all under my specific fork. Okay. So yeah.
So go back to the So going back to the graphs.
Yeah. So my script just lists all of the peak voltage, peak current values, and plots highest, lowest, and peak absolute current. And as this is the calcium channel, this is basically equivalent to figure 3C, I think it is, from the Boyle and Cohen paper. It's just upside down.
Oh. Okay.
I'm pretty sure that what's happening is that we're just looking at the opposite direction of opposite polarity of current flow. Because if you invert this graph, it's pretty much exactly equivalent to what we're seeing in the paper. I did have a spreadsheet showing it somewhere, actually.
So this is the other thing here, by the way, just to take a step back for folks. So again, another advantage of having the model that is described in this declarative form is that now we can build these tools that generically are going to analyze any channel that's described in that way, and will give you this plot. Whereas if you have it embedded in some program, some Python program, then you can't just run any program on it, right? So what all these tools now from this point forward here are they just take the input, like, okay, what channel do you want me to run on? Okay, I'm going to run on the CA Boyle channel. Okay, great. Here's the IV plot for it. So I just want to sync this point here because as like Wilson and Vahid have been looking at the ion channel spreadsheet and have been thinking about how to basically take papers and start to like take an ion channel that we want to have for a motor neuron and make a description of it. What we want is to, number one, make it as the NeuromL file that has the ion channel, and then number two, run it through this tool chain and have this tool chain tell us the current voltage relationship rather than having to write a custom script to show it to us every time, or having to take the parameters out of the NeuromL file and plug them into a Python file. If we're all working under the same sort of library of NeuromL files, we're going to very quickly see, I think, an explosion of collaboration that we can do around this sort of stuff, and it'll be much more verifiable and all that good stuff. Okay.
Okay. Yeah, I just pulled up this spreadsheet where I basically invert the values we get here and plot them. And yeah, this is basically figure 3C from the paper replicated. And yeah, it's basically the inverse of this.
Awesome.
Okay, so have I
think so.
Covered everything about how to get to where I've got to with this?
I think so. Are there questions though from folks here who've been listening patiently? Okay.
So I just had a question on the different scales, because I saw that in your computer you have like picoamps, but I think it's another unit, right? It's like amps. I know it's a minor detail. Just want to know why the numbers are smaller. to the scales that are in the paper, because in the paper we have like milliamps. I'm not sure why it's appearing as a 10 to the minus 12 or 10 to the minus 13.
Ah, okay. Yes, yes, all of the values I've got as output here are in amps. I mean, we could convert these to to microamps or picoamps. Whatever
Okay.
is in whatever's in the paper. The actual values should be the same, I think. It should just be a case of
it's like a scaling issue. Okay.
Yes, it would just be a case of converting the units.
Okay.
Yeah, we do want to resolve those things. Because they do matter.
Yes, that's probably worth doing just to make it easier to check that they are exactly the same.
Exactly.
Yeah.
Okay. Good. So all this stuff here is the stuff that's like kind of harder to run right now, as evidenced by the fact that sort of only Vahid has been able to run it at least a little bit. I think, unless somebody else here has been able to run this complement. Oh, okay. Joe can run it too. Okay, great. So good. So some folks have been running it. So that's great. So we want more people to be running it. So this is the modeling sort of technique that we want to be using, is writing up ion channel Hodgkin-Huxley files, or just so that everyone's aware of what those are. From here, this is exactly the same technique that we're going to use for motor neurons, for every neuron, basically. So all the neurons that are in this next thing I'm going to show you, which I only have 10 minutes to show you, they're all also encoded in NeuroML2, and we want to be able to describe the ion channels for those when we get to the neurons. But because we had this model that was already published by Bowen and Cohen of the muscle, the body wall muscle, it was electric, that had channels in it. It seemed like this perfect little prototype. It's only got 4 channels, so it's very easy to kind of understand what they are, but it's a place where we can see this entire tool chain all the way out to IV plots working. So the idea is that if we can learn this, and we can also have our repo well described enough, have good enough documentation, make it easy for somebody else to come in and learn, right? The challenge is that we've got a whole mess of other ion channels that we don't have NeuroML files for. We don't know exactly what neurons they all go to. But we need to match them over. So the idea is to take this enterprise that we've done on this little example of a muscle model and bring it over there. As well, the synapses between the motor neurons and the muscle cells and other synapses are also going to be described using NeuroML2. The gap junctions that are between the different muscle cells or between the different neurons are also described in NeuroML2, also have IV relationships. So all this stuff that we're doing here just kind of goes to the entire rest of the project when it comes to the neuron modeling, and so that's why this is kind of a core thing. All right, Travis asks.
I have a question.
Okay,
so
go ahead.
basically, we don't have all the experimental data for all the ion channels, right? And we only have 2 or 3. So we want to create first the working model and then somehow acquire the data from further papers or from experiments.
Right. Well, so the situation for ion channels is better than just 2 or 3 in the literature. Okay, so there's this aspect of mining the literature. I've got at least 10 or 12 papers of different ion channels that have IV plots in them for channels of interest in C. elegans. And
okay.
So I know this is the challenge with the spreadsheet stuff, which is how far to go. So I'd say I think I have at least about 10 or 12 for different channels where we have the current voltage relationship, and there are sort of different ways that we can infer our way into where those channels go, into which neurons they go. Some places it's more perfect than others. The trick is basically to say, like, we want to find those areas that we have the most knowledge and build out our model there, right? So where there's well-studied motor neurons, we want to flesh those out. Where there's well-studied particular ion channels, then that particular ion channel, we want to put it everywhere we know that that ion channel would be in the model, that kind of thing. So we want essentially our model to reflect the state of knowledge in the biological field right now, even though knowing that the state of knowledge in the biological field right now is incomplete. But we want our model to reflect all that, all of what's there and what's not there in an integrated way. So that's the key thing, is that by putting into the model, we now see exactly where the gaps are. Nobody else is trying to do this. Nobody else is trying to fit this puzzle together so that they can say, yeah, actually this paper said that this ion channel is in this neuron. Or, we have a general idea that this ion channel would be in that neuron. And if you run the dynamics of it, here's what it looks like. And like, okay, so is it right or is it wrong? Well, likely it's wrong at first, but then there's a paper trail which shows you what that where that is. And so from there we start to build up a resource that biologists will be like, maybe ideally down the road there is a new graduate student coming into C. elegans who doesn't even know how to program, and they're like, hey, the OpenWorm model needs to know what are the channels in AVAL neuron. Okay, and a grad student could do the experiment. It's actually not that hard to do in C. elegans. So the motivator here, what we're trying to create, is to have enough of a credible model that has enough of the pieces put together, exposing the gaps very clearly to the whole community of the C. elegans researchers, so that to motivate them to go further. But yes, there is still more for us to do, because there are still ion channels that have been identified in some neurons, and we do have some properties of those. So I'd say that, optimistically, how much of this information do we have about ion channels and neurons? I'd say maybe it's like 20 or 30% of what we really need for the whole connectome, right? So we know it's not going to be fully complete, but with the first 20 or 30%, putting it all together into a computational model will be the first time that anybody's ever done that. First time any group has ever done that, and we're talking about an organism that we've had the connectome for like 30, 35 years for, and no one's ever done that. So it would be a huge improvement over what's there, and it would show the community, what kind of needs to get done. Because right now there's like not a motivation to go and like, oh, what is the ion channel for that one random neuron, right? Like for some PhD student to spend his time doing that right now is kind of like, well, why would I do that? Or they don't even know like where we know things. They don't even know where the gaps are. So there's not been a systematic approach to like every single ion channel, every single neuron that I think is really needed. But so far that hasn't happened. So yeah, that's kind of the idea behind this. I know that it's a little, from the computer science perspective, it's sort of annoying that there's not like a database that has this information that's like already there. Like I would have expected that there would have been, but there isn't. And so we're making that basically database, but we're making it as a dynamic model, because if we didn't make it as a dynamic model, we couldn't test these properties. We couldn't compare our IV curves to the ones that are really there and that kind of thing. Make sense?
Yeah, that makes sense. That's our mission statement, I guess.
Yes, exactly. And I probably need to find a way to like write that down, because I think it's a subtle point. Okay, hang on. So some questions that got typed in chat. Okay, Travis asks, so Rayner's graphs are the ones we will be validating against the digitized Cohen graphs. I
meant
Yeah.
when we go to optimize our cells to be like the Cohen graphs, we're going to be comparing. I'm talking about IV graphs now. We're going to be comparing our IV graphs. They'll be made with a script like Rayner's, something analogous to that anyway. And then we'll be comparing those graphs to the digitized Cohen graphs. Is that what's going to happen? I just want to be clear.
Basically, yes.
Okay.
Yes. So the whole optimization thing is I actually need to kind of brush up on exactly which of the libraries that we're using there, and I think Miguel has started down this path a little bit. But I think behind the scenes, hopefully, we will actually be using the digitized graphs to basically pick the right parameters to make them. So so they will like not just validate, but like define essentially.
Okay, yeah.
Even though we know that there's a lot that's unconstrained, and you could probably pick the parameters in many different ways to get the graphs, which is another one of the weird things about biology. But at least picking some parameters that are within biophysical biophysically realistic dimensions is an improvement. So yeah, that's basically right. Yeah.
Okay, thanks.
And I think IV Analyze itself can be improved, obviously, like sign flips and stuff like that, so that we can get even better. But yeah, that's basically the idea. Yeah. And then Joe says, so I'm going to volunteer to work on the Cloud DB interface. I want to get mostly finished with movement verification one first. I'm thinking integrating all these tools so uploading graphs can be used to generate and test whole models for REST API access. Okay, cool. Yeah. I think the cloud thing will add some levels of convenience if you think you can do it quickly. I think there are also these sort of like basic things that folks are in the tool chain to make happen. But I guess I think you're probably thinking about it in a way that this doesn't seem like a long, drawn-out process, right, Joe? So something relatively straightforward to do, because you're already kind of doing it on the movement validation side. Oh, I can't hear you. Oh, we lost your audio somehow. I can see that you're going off mute, and can other people hear him? It's not just me, right? Okay. Oh.
Anything?
Yeah, there we go. Now you're back. Don't know what happened.
Now I'm back. So that works?
Mm-hmm. Yep.
Okay. So what I was saying is that kind of the direction that people are going? I mean, my thought was to be, I mean, in the end, kind of the end view of things would be something where you'd take a model that a researcher had lost the video. Do I still have audio?
Yeah.
Okay. So taking like a graph, uploading it to the site, it does the computations, it figures out the whole, using a lot of Reiner's models, and software just kind of behind the scenes to generate a model, and then we can have a kind of a central database of everything, along with what we all currently think is the right way to do it.
So I think that's a great idea. I think I've been focused so much on the scripts that I haven't really been thinking about this as much as a service, although I do know that, yeah, the movement validation guys are thinking about it as a service too, so maybe it makes sense to have in parallel. So I'd say, yeah, give it a shot. It certainly beats running all this stuff on the command line.
Yeah, and to be honest, I'm always looking at boosting my Django experience on my resume, so
cool. Yeah.
all the more.
Great. Okay. So that sounds great. Okay, we're out of time for this session, and there's some things we didn't get to, specifically relating to C302, which I want to do an intro of. So I think we're going to leave that to next time. And I know some folks, at least Abdullah here, who's joined us, has been spending more of his time on the C302 stuff. So thanks for being patient through this session. But I think, what he has done is he's looked at the NeuromL2 implementation of C302. Basically, C302 is NeuromL2, a simplified NeuromL2 applied to not just a muscle cell, but the whole C. elegans connectome, and it has all the connectivity in there. Now, it is the thing about C302 though is that to date, the model that's used for each neuron is essentially a Hodgkin-Huxley model. Meaning that it spikes, that all the neurons spike in C302, that they have action potentials. However, C. elegans neurons in reality do not have action potentials. They do not spike. They don't do that thing. So using a Hodgkin-Huxley model is a gross oversimplification when it comes to the dynamics. Okay, they just definitely do not produce action potentials. At least no one's ever shown any evidence of them. The muscle cells actually do produce action potentials, which is kind of the interesting thing about C. elegans. That said, NeuromL2, if you have the right set of ion channels, will produce a non-spiking cell that is just an analog cell with just exactly the same, all the same infrastructure, but just with different parameters for each ion channel. You can make a neuron that doesn't spike. And so what we're going to be getting into is reproducing these, 6 or 8 motor neurons next, which are the motor neurons that we know connect to this muscle cell. We picked a specific muscle cell a long time ago, a specific body wall muscle cell, MDL08, and we know, because we have the connectome, which are the 6 or 8 motor neurons that connect into it. So the idea, and this is really what we've been wanting to do for a couple of years, and it's just taken us this long to kind of get there, is to start with a simple system of the motor neuron plus those muscle cells and the synapse between them, and describe all of that in NeurML2 with as much as we can get the right ion channels for those motor neurons and get the synapse right. And we actually have a neuroconstruct project of this, which I haven't told you anything about neuroconstruct because it's another tool to install, and some of you have played with it. But there is a neuroconstruct project that has all 6 to 8 motor neurons, has the muscle cell, some basic synapse, but again, it's using these very simple dynamics for the neurons. So just as an advertisement then, because we didn't get to C302, but I did want to say something about this, scheduling the Synapse Paper Journal Club. So what we've started to look at is this next jump. So once you have a muscle cell that's relatively well defined, that matches what we know from Cohen and Boyle, which by the way also has its own problems dynamically, but we'll leave that aside for a second. You want to describe that synapse, okay? The synapse where a neuron connects to the muscle cell to make it move. And there's a paper on that subject, and we are going to have a journal club on that subject in the next 2 weeks or so. I'm going to send out a date for it. It'll probably be on a Friday. It'll probably be like in the morning in Pacific time and in the evening in GMT time. And we're going to walk through, it's just a biology paper. It's just a biology paper talking about that. And this is where Rainer and I actually started about 6 months ago, and then we kind of came back to the muscle cell model because the way that this paper works is it basically shows you how the synapse, the dynamics of the synapse by recording from the muscle cell and seeing how the muscle cell dynamics change relative to stimulating these motor neurons that connect to that muscle cell. So we're going to go through that paper. There's actually some folks more on the biology side volunteering to help us work through the heavy genetics content of that paper. So we'll learn a little bit about that. We'll talk about optogenetics in that journal club, which is taking ion channels that are controlled by light and using special frequencies of light. Yes, I will send out the Synapse paper link. But using special frequencies of light to do it. In fact, it's linked. The Synapse paper is linked on the agenda, Miguel. The link on the word Synapse paper is the paper, so you can get it right now. Anyway, and so through these optogenetics experiments, which are also really cool, they can stimulate 2 different kinds of motor neurons. They can either stimulate the ones that have GABA or the ones that have acetylcholine, and they can see what the impact is on the muscle cell. So here's a place where we don't have a given model of a synapse, but we want to make one. And it's important for us to understand the underlying biology so that we can know what we're fitting to. We haven't really done much fitting of any synapse of any kind. So the other reason this is important is that whatever we do here for this synapse, we're going to reuse for any other synapse, any sophisticated synapse that we have anywhere else. Right now we're using very simple synapses for everything, so it's we know it's not super accurate, but this is the process. So I just wanted to advertise for that. You will see it go out on the mailing list. I will post it on the chat room. I invite all of you to come watch it. Not everybody needs to be in the room to see it, so I will put a call out for folks who actually want to be in the room to ask questions if you're super interested. And then when we get to the end of that process, we will have a better understanding of what biology we want to target, and we can spin out this conversation to getting that done. So, all right, let's see. So Joe says, so even though most of our synapse models and our neuron models are poor approximations, but we're still already getting results out. So what results are we getting out, right? So again, this is a bit of the philosophy of the project. Okay? We are getting results that show us what the math says the output of this system of equations is. And we're fitting piece by piece. We're fitting the ion channels, fitting the whole cell, right? We're fitting the synapse. So we're fitting all of these pieces as we go to what we see in real papers. The results that we're getting out are, you can run a simulation with any number of parameters or any values for any parameters, you're going to get something out. But we don't claim that those are biologically realistic until we can point at a paper and say, yes, this is, this is what it matches. And there's going to be some places where we don't have any data at all when we get aggregates of dynamics. But again, as I was sort of saying with the ion channel idea, nobody has even done this much. So everybody is operating in the absence of any model that's any good at all and that's based on any kind of biophysical reality. So some of the things in terms of like running the whole connectome, which you can do right now, you can run C302 and you can see some dynamics, but now that you've seen what detail looks like, I think you can start to appreciate how off those dynamics probably are, really. So it's just an abstraction of running a connectome, and we've tried to be very transparent about the fact that we don't think that the dynamics in like C302 really reproduce what's happening in the actual worm yet. We have lots of reasons to know that it's not what the worm is actually doing, especially when you have Hodgkin-Huxley dynamics for all the cells, because we know they don't spike. But that said, there's interesting things that come even just from doing that. But yeah, like a C. elegans scientist is not going to look at that and say, this is going to give me great insight into how the nervous system works, because we know they don't spike. So we have to keep moving towards where the biologists are, essentially, and making the model better and better. And that's the real tipping point here is when biologists come to this project, don't have coding experience, and they get more and more useful knowledge coming out of these simulations. That's the tipping point where I think where this thing gets really even more exciting in this next phase of the project, where not only are we reproducing biological activity, but we're doing it with this very rich underpinning of real data. Okay, so I've gone 10 minutes over. Thanks, guys, for hanging out. I usually try to keep this right on time. So we will take the other agenda items, like the intro to C302, and do that for real next time in this session. The Journal Club will be a separate thing. Thanks all of you for the contributions up till now. It sounds like we got plenty of sort of next to-dos that have come out of this. Let's take it to the chat room to get more specific about those things, create issues for stuff that we talked about here. We'll aim to do this again in about another month or so. Also, there will be a general meeting next Wednesday for OpenWorm. I just wanted to make one comment about that. Sorry, I want to get out of here. But one comment about that. This sort of general meeting is like this, I don't know, tries to wrap up everything that's happening across the entire project, and just the way that things have happened, there's just too many contributors to all fit in a single hangout all at once. So like, yeah, this is like a limit, I think, of like 15, I think 10 for these kind of hangouts, maybe 15 total. So we just can't fit everybody in a single room. But that meeting is going to be looking across everything that's happening, looking across movement validation, looking across Geppetto, looking across this stuff, looking across cybernetic stuff. So I can't invite all of you to it just because you don't all fit. But you are always welcome if we have a spot to join and listen and contribute. Otherwise, I'll just kind of be providing the update to this, and Rainer usually joins as well. To kind of just give a sense of what's happening in this space. But that's also public, and that's a public hangout. But just so you understand when that's running, if you see this meeting and you're like, oh, why wasn't I invited? It's not because we don't like you or anything like that. It's just purely because, like, yeah, we just can't fit everybody in. So we're gonna try to do that meeting every month, and we're trying to do this meeting every month, and we're trying to kind of like report in on what's happening and that kind of thing. So feel free to watch and see that meeting and get a sense of the bigger picture as well. But this stuff here is really important separately. So. Okay, so thanks everybody. Let's take everything else to chat from here on out, so we can get everybody out. Happy New Year, and talk to you guys all later.
See you guys. Thank you.
All right, see ya.